Hi, I'm Jim Allison. I have the good fortune to, along with Andreas Schattinger, Pam Sharma, and Tony Rebus, of organizing the 2024 meeting on tumor immunology. The four of us got together and discussed what we wanted to cover, and one of the things that was driving all of us, I believe, is that it's been 11 years, 13 now, since the approval of ipilimumab, the first immune checkpoint blocker in the treatment of cancer therapy, the dawn of this new era. What we tried to do in this meeting that we organized, the four of us, was to try to look, 10 years, 11 years post-IPI, where are we? And where we go next? That's why we have a session, for example, on failed trials and what do we learn from them. We know that, as long as you don't have toxicity, you could give a half a dozen different checkpoint blockers or agonists. You have to know the mechanisms and know how they're going to work together. But I think that's the only way we're going to get from 60% or 55 to 60% in melanoma up to 85 or 90%. That's the only way we're going to get in bladder and kidney cancer from 30% or 40% up to 70%, 60, 70%. And and the fact remains is, again, after all this time still, we don't have much purchase on glioblastoma or pancreatic cancer. And these are things that we also need to approach it. And I think, there have been some combination trials using radiation, chemo, and some checkpoints that use as many as 6 different agents in some of the new trials in pancreatic cancer. They're beginning to see some hint of response. So the precedent is there that we can combine many things and should be open to combining them now. But with the advent now in the last couple of years, and with the amount of data that's coming out of these neoadjuvant trials, we ought to be seeing more evidence of these things. And I hope that will be reflected in the talks that we have at our meeting, and where we can get together there and start talking about which of these should we put together to move the field forward and realize the curative possibility. That's the main thing. I think what sets this meeting apart is that we are trying to get both people that have had a lot of clinical experience, so we're going to attract more clinicians to the meeting, but also people who are doing basic studies, basic biology study, and in preclinical studies in animals, or even in tissue culture, to understand the mechanisms of these molecules, of what I would call discovery science with translational science, with clinical science, so that we can again bring them all together and hopefully accelerate the movement of new ideas into the clinic, and more importantly, to FDA approval. What I'm personally most excited about is I think what we're going to be seeing from the new Spatial, ability to do deep dives into omics, transcriptomics and proteomics, and build it into a spatial idea of what's going on in the tumor. We can really more quickly identify cell types that are that organize structures within the tumor. I hope that scientifically there will be more data presented about the importance of interplay of these different pathways, these different molecules, if you will, in an overall immune response. And then secondly, how that can inform clinical trials, either sequential or concurrent application of different agents, depending on what we learn from analyzing tissue specimens from the neoadjuvant trials now that are clearly the dominant thing that's going on in the field. I hope the clinical scientists that are attending the meeting will get some idea of the fertile ground for advances there are, and perhaps help influence the people that they can talk to at the pharma companies and the biotech companies. I think fellows, both students and particularly postdocs that are just beginning to get their feet wet in these areas, can come here and actually be exposed to people, more senior people that are doing things maybe at a high level, but also mid-career people who are learning and maybe asking some questions the more senior people aren't asking anymore. They're based on new biology and ideas, and having a place for it. I know that we made an effort to both include a lot of mid-career people who are, with their enthusiasm and vigor, their freshness in the field, but also leaving open slots for fellows and things that through the abstracts have interesting stuff that they can get up and tell everybody about. One of the services that the Keystone meetings provide to the field, I think, is a place where people intensely engaged in research can get to exchange ideas. It's a place for fellows to learn and get ideas and network. Something that I think about more and more is it's a place where if you're building a program or thinking about hiring faculty, it's a wonderful place to come and see not just the senior folks, who probably aren't going to go anywhere, but see these people that are just beginning to come out and have a chance to hear them talk, sit down and talk with them for a while, and help you recruit. It's one of the reasons I come to the meeting.